first I'd like to thank the organizers for inviting me and to give me the opportunity to talk to you about the work they've been doing with vocal learning birds and some comparative data that show a little bit about humans as well. I'm not going to be showing any much fancy imaging, but in fact, I'm going to start with a general introduction to the field. Then I'll focus on presenting a database that we've been developing, which is analogous to the Allen Atlas. We are developing a brain atlas of gene expression in zebra finch, and then I'll highlight some of the contributions that this resource are given to us, to the field. So I'll start right away with a clip, which is very illustrative of a day of life in the zebra finch family. Could you play the clip, please?
A juvenile zebra finch is about to be exposed to the song of an adult male. A female is introduced into the chamber, and in response, shortly thereafter, the male begins to sing. The juvenile, 4 to 5 weeks old, is paying close attention. Like a human baby learning to speak, the juvenile will try to recreate the sounds until he gets them right.
So I really like this clip because actually it's not easy to get this, and the crew from Oregon Field Guide was able to get really nice filming. And by the way, that's Tarcísio introducing the female to the chamber. And also we experienced some sex and song in that clip. The male actually tried to mount the female very briefly. It was not very successful. But that illustrates a couple of things, that the song is used in courtship context in the zebra finches. It's a dimorphic behavior. Males present it, females don't present it. And the juveniles, the young males, have to learn the features of the song in order to be able to sing and to be successful at attracting mates. So the learning, this is actually adapted from some work from Alison Dope, showing that here the motif, that's the song, the stereotype song that the adult male sings, has a set of syllables with a particular structure. Both temporal and spectral features are very conserved. And the juveniles start almost like babbling, what we call babbling a sub-song. They have very immature vocalizations that gradually shift into a pattern that eventually matches the pattern that they heard from the adult. There are two major phases to this process. One is hearing the sound, being able to memorize the sound, and later imitate. The imitation is done through sensory motor processing that requires sensory feedback. You need to hear the sounds that you're producing and make a comparison between the sound of the vocalization you're producing with the sound that you internalize, memorize from the adult tutor. And based on a mismatch, then your motor control pathways modify the song output so it gradually will match the model. This is pretty amazing, remarkable feature. That's the basis also for acquisition of human speech. And in fact, the speaker yesterday, Pat Cole, alluded to some of those similarities. And some of these, interesting. There's an old version of it, but so of the talk. But this is adapted from a review by Dope and Cole, highlighting many of the similarities between the two models. Both need an auditory model, that is the song of an adult. Critical periods are present for sensory acquisition. This memorization needs to occur within a certain window in the life of the birds. There's need of auditory feedback for practice. The babbling, which is the immature vocalizations in the early phase. Individual variability in vocalizations, dialects, and so forth. There's a phonological structure that needs to be learned from those calls. This behavior is very rare. The ability to mimic this vocalizations you hear, of course, is present in humans as the basis for speech, for spoken language. There's good evidence that's present in whales and dolphins, cetaceans, and in bats. Maybe some anecdotal evidence in a few other mammalian groups, but that's fairly rare. but remarkably it occurred in 3 groups of birds. And I want to emphasize there are over 30 orders of birds, and only 3 orders evolved this ability. This is the songbirds, the parrots. We know parrots are very good imitators, and hummingbirds. Many people do not know, but hummingbirds have complex vocalizations, and they are learned. They have dialects and so forth. We actually study them quite intensively in the lab. So I want to point out that even though it's only 3 groups, songbirds are responsible for include more than half the whole number of species in the world. There's roughly estimated to be 8,000 to 10,000 species in the world. Probably about half of those are songbirds. So even though it's one of the 3 vocal learning orders, it's the most specious order, most diversification, possibly related to the ability of them to have vocal learning. So I said it's rare, and it's not present in rodents or non-human primates. By this, I mean the ability to hear a model and imitate that model. In fact, OK, I guess some of the slide that I want to show is probably not be here. But OK, so that ability is not present for the most part to the rodents and non-human primates, even though they have the ability to learn the contextual use of vocalizations, of course. And there's some evidence of some ability to modulate some acoustic features, but there's no evidence that monkeys, for example, have dialects or that they are capable of hearing vocalizations and then learning to imitate that pattern. And in rodents, the evidence that you can cross-foster, you can deafen, you can even ablate the telencephalon, and they are still able to produce vocalizations which are largely unchanged. So I hope to convince you that this is not a good model to study learned vocalizations, the process associated with learning to produce your calls. So we focus on birds and songbirds, and this is a schematic here showing diagrams of the avian brain. This is a zebra finch brain. This is to scale here to the human. To a large extent, the basal vertebrate organization is conserved. But very importantly, for most of the 20th century, it's thought that most of the avian brain was basically an expanded basal ganglia. And that was because largely in the avian brain, you don't see a cortical layered structure. And birds were thought to be dumb, to be only capable of very unlearned behaviors. This bird brain concept. But by a lot of work, including connectivity, molecular analysis, and cytoarchitectonics, we now know this concept is wrong. There's actually a small volume of the telencephalon is actually basal ganglia. We can find striatal, globus pallidus regions. And then the majority of the volume of the telencephalon has a similar origin as the cortical areas, as well as amygdala and claustrum, some of the structures like that. And with a different cytoarchitectonic organization, but it's definitely more paleocortical-like rather than basal ganglia. And this is many insights were gained by work by Harvey Carton and others. The idea is that many of the different cell populations that are in different layers of the mammalian cortex are present in physically displaced segments, particularly some of the portions of the dorsal-ventricular ridge in the avian brain, the sauropsid brain, that releases off this type of representation. And importantly, the insights that were gained from a lot of connectivity studies that the elements in different cortical layers are physically separated, segregated physically in the avian brain. But we can find, this is an example from the avian auditory pathways. You can find thalamus-recipient zones, and you can find another area that sends the same projections in intercortical circuitry, and these are segregated. There are a couple of implications here that possibly in the avian brains, the fact that these cell population layers are separated facilitates the study of their analysis. And also the idea that there's not only one way to organize your higher cortical circuitry. You can have very similar functions. In fact, in some cases with higher acuity even, with a very different organization. And this is a very fascinating topic. We are really interested also in the determinants of the cortical layer organization versus this more nuclear organization. But so there's practical implications and more important fundamental implications. This is the circuit of the vocal learning circuit. I'm showing here the direct vocal learning pathway that includes this nucleus HVC and array. These are cortical structures. RA is the main corticofugal pathway, and it projects to respiratory and vocal nuclei in the brainstem. So a couple of things I want to emphasize here. These brainstem vocal and respiratory nuclei are very conserved throughout vertebrate phylogeny. Fish, the vocalizing fish have similar things. Non-vocal learners have vocalizing centers. The midbrain is presumably here in the bird analogous to parts of the periaqueductal gray in humans and mammals. And here the M12, which is the 12th nerve that innervates the syrinx, this vocal organ, is analogous to the ambiguous nucleus in humans that innervates the larynx. This is direct pathway for vocal production, the production of vocal learning signals, and lesion of any of these nuclei eliminates the ability of the birds to sing, the learned vocalizations. In addition, there's a rostral pathway that includes cortical basal ganglia, your area X, thalamo-cortical loop, that's essential for vocal learning. A lesion of any elements in the circuit eliminates the ability to acquire the learned vocalizations. So these are different pathways, and they are somewhat easy to study in terms even of electrophysiology. You can put electrodes and record their activity during singing and so forth. These nuclei, the forebrain nuclei, are absent in non-learners. So the majority of groups, such as pigeon, chicken, quail, many other groups that are not vocal learners, they absolutely lack these nuclei here, even though they have the brainstem nuclei shown here. Yeah, actually, this is the slide. Just to convince you that a mouse, even that lacks this genetic manipulations, that lacks telencephalon, is still able to produce those ultrasound vocalizations to a fairly abnormal extent. And this would absolutely not happen in a songbird. If you don't have the cortical basal ganglia nuclei, you are unable to sing as a songbird. Some early work in my lab, and Eric Jarvis, my colleague and collaborator for many years, we used early gene called zinc to map auditory and vocal control pathways. This just shows that this is one of the early examples showing that you can actually, the birds, when they're communicating their hearing, there's high induction of this immediate inducible gene. This is one of the early examples of a natural stimulus, of natural induction of these inducible genes, and it was very useful for us to map the auditory system of songbirds. And Eric also similarly showed that you can get activation of the basal ganglia cortical nuclei, and these are context dependent. It's a really interesting story. But just to make it short, we use this tool to map the vocal control systems in all these vocal learner birds. A lot of this was already known in songbirds. Some of the pathways were known in parrots. Nothing was known about the hummingbird. And we actually were able to do this. Studies, we actually did this in a tropical forest in Brazil. And by capturing birds that had been singing shortly before they were sacrificed, we looked at the expression of these early genes, and we found we were able to define auditory and anterior and direct vocal motor pathways. And what's really remarkable is that these are different groups, and they evolved very similar nuclei with some differences in the position of the brains. But I want to remind you that the majority of the birds are non-learners. So these are events that occur independently in evolution. Now, this slide is already outdated, and the reason is there's a very recent effort that we were part of, largely coordinated by Eric Jarvis at Duke and Beijing Genomics Institute and the Smithsonian and several other groups. There's a very large effort to sequence the genomes of birds. We've sequenced over 50 genomes, the largest number of genomes in any vertebrate group currently available. And a lot of insights were gained from this. As a whole issue dedicated to the topic was published in Science at the end of 2014. And I won't go into detail, but whole avian phylogeny was modified. It was pretty amazing, because you think phylogeny and bird phylogeny is well established. It was actually quite remarkable how there was a lot of convergences of avian traits. So this tree is a complete revamp of the old established phylogeny. It's pretty remarkable. Anyone interested, I highly recommend go and take a look at the articles here. For our purposes, there are two consequences of this reorganization. We now think that parrots and oscines and songbirds had a common, our sister taxa with a common origin, probably in Australasia. So this is true. Probably vocal learning, this parsimonious explanation, the common ancestor to parrots and songbirds was a vocal learner. possibly with a loss of the trait in those intermediate suboceans. So now we are thinking this attacks of a common origin for vocal learning, but very importantly, hummingbirds is a completely separate group that's really now well established. So all the features I show you, the hummingbird vocal system is a system that arose completely independently of the songbirds. And we have opportunity here to look at convergent systems that evolved completely separately. Before I describe the database of gene expression, just want to point out that that's been really remarkable advances in looking at the genetics of vocal learning features. And in fact, there's a discovery of FOXP2 involvement in humans in a severe speech disorder, a language disorder, which is speech dyspraxia. And this is the autosomal dominant disorder that's transmitted, and it was shown to be associated with FOXP2 gene transcription factor. There are a couple of mutations leading to this disorder. And the Constance Sharp group was able to show that if you knock down these FOXP2 in the striatal, in that area X of the striatal, the vocal learning pathway, you disrupt the bird's ability to copy a model. So it's a remarkable example of similarities in molecular mechanisms underlying acquisition of learned vocalizations, both in humans and songbirds. And I'll go back to some of more other similarities a little later. So now we have two questions that we want to address by looking at more gene expression patterns in songbirds. One is to get more into the molecular determinants of vocal learning and the properties of the related circuits. And the other broad question is, are there molecular correspondences between avian and mammalian brains? Because I show you, even though they have similar origins, the organization of the adult brains is quite different in terms of cortex and the avian telencephalon. Are there convergent patterns, and can those patterns help us resolve issues of homologies? Which areas are homologous across birds and mammals? So we developed this project. We started with a small pilot looking at a small set of genes that were enriched in song nuclei, and we quickly noticed how interesting the patterns were and how they were used in a complementary fashion. So we decided to apply for funding, and we were able to, first through the pilot and then through a resource grant from IGMS, to map a large number of genes in the brain, starting from markers of the song nuclei. And we developed this database that's now currently available. It's very useful for us and many others. And so this is the website. We currently have completed patterns for 500 genes. We have another 500 in progress. So we have processed around 1,000 genes. That's like 1/20th of the genome with the expected number of genes. So it's a fraction of something that you see in the mouse atlas. But we think these are really interesting data sets for here. You can find these genes listed alphabetically by markers and gene families, and I'll show you a few of these features. The database, individual entries, there are links to several genomic features and links that like ensemble and entree genes, and that show you a little bit of the structure, and also the UCC browser. You can look at the structure of the genes, the references, and the probes you use to probe these in situ hybridization. But more importantly, the actual database consists of sets of sagittal serial sections, providing with in situ hybridization. We put a lot of effort into optimizing the protocols to minimize background, and really proud to say that this is really high quality in situ hybridization with these base probes. We published this recently, the optimized protocol. These is like images that you can navigate. They are scanned at high resolution. In fact, by our collaborators, Partha Mitra, Cold Spring Harbor, who has helped us set up this the scanning and the analysis. We developed something that was also missing for us, which is a high-resolution histological atlas, again based on Harvey Carton's histology and Partha's imaging. And in my lab, we have done the annotations of all these areas, which is a heroic effort because there's a lot of structures that were basically in uncharted territories. And apologies, it's a little dark here, but I want to show you is light, but I want to show if we can zoom in and look at high-res images, we have really high quality in situ data. And this is an example of one marker of one of the song nuclei at HVC of a marker that's enriched in this nucleus, and in fact is enriched in one specific cell population, which is the striatal projection cells in this nucleus. You can find, there's over 200 structures that you can find markers of various areas, and then you retrieve the outputs by genes, and you can find markers of specific nuclei, also the matrix indicating what other structures they are markers of. And these are just some examples to illustrate how we have all kinds of patterns, really nice markers of several of these song nuclei. This is striatal nucleus. This is the premotor nucleus. And some are positive markers, other kind of negative markers of this, and various combinations. And here also delimiting what might be some equivalent of some cortical fields. These patterns are then quantified by densitometry, and we are now in the process of generating molecular signatures of these various subdivisions with the idea of then comparing with the data from the island, for example, and look at what might be the equivalent, molecular equivalences between this. So these are the collections of genes that are in the zebra, markers of the song nuclei, genes of physiologic relevance, various neurotransmitter types, markers of major subdivisions. And now I'll highlight a few points here that we have learned from these. There's some remarkable cases where by looking at these patterns and looking at gene expression, looking for markers of cell types and cell groups, one of them that's really surprising is that we noticed looking at the genomes that the dopamine transporter, the DAT, basically, which is thought to be, as in textbook, that is a highly conserved gene, is actually missing in birds, not just birds, but birds, sauropsids in general, lizards, turtles. This whole entire group lost the gene. The gene is absent. So dopamine transporter is not a fully universally conserved gene. This is very intriguing because, in fact, there have been people who have shown that there is dopamine reuptake activity in the avian striatum. It turns out that by looking at the molecular indicators, here we're comparing with the mouse data from the island. Instead of NAT, because they have expression of NAT, the norepinephrine transporter is expressed in the substantia nigra, and that's quite remarkable. It's not at all the case in the mouse. And so you have a case where the loss of the dopamine transporter results in the expression, the compensatory expression of NAT in dopaminergic cells. So this is unique now to birds and lizards, and absolutely not in the mouse, and is not in fish either. So this is not an ancestral condition. This is a condition associated with the sauropsid lineage. This is double labeling to confirm that these expressions not just in the nigra, it's in the dopaminergic cells. And of course, the control shows that the NAT is expressed in the norepinephrine cells of the leukocytes as expected. A couple of other surprises, and applies this is really light here, but the VGLUT1 is a major marker of the cortex. We were naively thinking we could use this as a marker for the cortex to get into ideas of homologies. In fact, this is a VGLUT1 in the mouse. This is VGLUT2, which is mostly subcortical. And in birds, actually, interestingly, the VGLUT2 is expressed mostly in the paleo fields. And we think VGLUT1 is either absent or it is highly truncated, and it's unclear whether the birds have VGLUT1. So this, again, another example of compensation, but also highlights the difficulties of looking at homologies with these molecular markers. But just to give you a different example, we can gain insights into this is a marker that's most exclusively expressed in the hippocampus and the medial amygdala, as well as perhaps a part of the piriform cortex in the mouse. And in the bird, we see here an example of really nice marker of the hippocampal formation in birds, and the tinea nucleus in the arcopalium of the birds, which was a candidate for the medial nucleus of the amygdala. So I showed examples where there's no correspondence, but we're really fun to explore and find these nice correspondences, which providing molecular evidence of possible correspondence of avian brain structures with the mammalian brain. So this is just some example of insights we gain. But I also want to show some data that look at the arrays of markers of these various nuclei. We were particularly interested in finches versus hummingbird markers because, as I told you, these circuits arose independently. And we are looking at the molecular markers and asking, do they use the same molecular mechanisms? And we find actually a large convergence of a subset of the genes. We're exploring now what are those mechanisms, because this could provide some insights into the basic features that are required for vocal learning systems. And in fact, we went even further collaboration with Eric Jarvis and actually with Lynn here from the island. We compared some of this transcriptome data and some of the data from our atlas with human data, and there is actually a remarkable correspondence. This shows is an index of similarity of profiling of multiple regions in a neutral way. So many regions were sampled in the zebrafinch and compared to the human transcriptome database from the island. And we were asking which regions have high correspondence. And some of the regions of the high correspondence turned out to be two of these song control nuclei, nucleus array in the vocal production pathway, and nucleus X in the basal ganglia in the vocal learning pathway. And this is shown here. So there is a high similarity in the markers that are differentially expressed in these nuclei, both in songbirds and in humans. It turned out there's a set of about 50 genes that are highly conserved, and here we've just shown one of the examples, which is something that's fully downregulated in the songbird array, and hummingbird, analogous of this nucleus, even in the parrot core nucleus, and is highly downregulated in the laryngeal motor cortex of humans, which is analogous to this nucleus that provides the projection to the N12TS. And again, the projections of the cortical projection to the medulla vocal nucleus is analogous to this area, the laryngeal motor cortex, that provides innervation to the ambiguous nucleus. It's remarkable to see that you have molecular correspondences here. So running out of time, I just want to show that some of the other remarkable markers of these are related to this. And by the way, that was a slit robo marker. I didn't mention that. But there are many other markers that are exquisite markers of this song system that are related to axonal projections. Many of these are involved in semaphorins and plexins, which are involved in axonal guidance and pathway formation, which we believe now is an important set of genes for the formation and maintenance of these things. This is an example of many family members related to axonal growth, fasciculation, and so forth. These are, we have NERPs, and semas, plexins, robos, uncs, and so forth. So we are doing an extensive characterization of these. And I have to skip this, but I just want to say one more thing, that another class of genes that were of high interest are ion channels, because they regulate neuroexcitability, have key physiological roles. And when we co-published the genome of the zebrafinch compared to chicken and non-learners, we found that many of the genes that are in the high strong selective pressure, which means they have high DNDS substitution rates. For those of you not familiar, this means that non-conservative substitutions are high compared to conservative substitutions in the protein code sequences. Many of these are calcium and potassium channels. And we examined then the family of potassium channel genes, and found that many of these are really exquisite markers of the several cell nuclei, including the vocal production and vocal learning pathways. This, we're particularly excited because this is a high, it's only expressed in large cells, and it's probably related to regulating its levels of excitability, and also the SN3B, which is another family, the sodium channel. This is more recent, showing that's remarkable development and regulation. This is a juvenile during the learning phase. Then it's downregulation of this SN3B. So we're seeing many examples of these. There are really nice targets now for manipulations, but these are candidates of genes that play key roles in the vocal learning process. And we're also looking at profiling cell types. This is backfields from the striatal and the cortical cells, and profiling HPC cells from an early study by Lombardino and the Nottebohm group, which we then have been characterizing these and looking at the AA localization of these, double labeling. One of these is one of the examples I showed earlier of the striatal projection. The other are markers of the cortical projections. These cell types have very different physiological signatures, and I want to point out, so we now have a collection of markers, a large collection of markers of these, and their associated promoters. We're beginning to look at their regulatory regions as well. But I want to point out that looking at the markers of cells like this is very exciting, particularly H52RA. These are have very sparse firing. They work from Mike Fish showing that they probably are associated with the encoding of features of the song. And these are also the famous cells that are replaced in adulthood that Nottebohm described associated with the seasonal plasticity in species like canaries. So this is the summary of all these different insights we're gaining from looking at this molecular database. And the next step is now manipulations, and there are some groups working towards developing transgenic tools and viral vectors. So I don't have anything to show today, but there's a lot of exciting advances in the field. And just here to acknowledge most of the people who have worked on the Zebra molecular database, particularly Pete Lovell and Morgan Whithlin, and several technicians and people who are working in the website. And Partha Mitra and Arvi have been instrumental in providing imaging resources in the Atlas, as well as our collaborators in the comparative work. And what I did not mention here, in that study I mentioned earlier, that Adeline from the ALA was also involved in that convergence study. So thank you very much.
We have time for a few questions. Yes?
Thanks, Claudio. That was very nice. Is there any evidence that the variance across birds in those who learn vocally and those who don't in relation to social interaction, the need, or in their environments? Yeah, it's very interesting. In fact, it's variable. The zebra finch is very social. And in fact, if you just present the sounds, that's not enough. It's clear that there needs to be interaction with the tutors. At the very least, have some model and motivation for the bird to initiate. So there are ways to try to improve that. But just hearing playbacks of the sound is not enough, suggesting there are many cues playing. On the other hand, I have to cite the classical example of sparrows that were studied by Peter Mahler and others. It seemed like in those cases, he took nestlings and presented just a tape-recorded song. So just the sound of the song of juvenile model to those juveniles was sufficient for them to imitate them a few weeks later. So there's definitely species differences. We don't know in terms of neural basis because the circuitry is quite similar across these. Also, some species are very good imitators of a species vocalization, some are not. And yeah. And what about the genes related to social interaction, as opposed to just the vocal skills? Yeah, we don't know much about those yet, but there's probably evidence of other circuits, like the septal areas and other areas involved in social interactions that will probably provide input to these circuits. So it will be very interesting to, in fact, the database we're generating is the whole brain. So we're going to be also cataloging the expression across these areas like the septum, the bed nucleus, and all these areas involved in social networks. And we will have that as well. Eventually, we could also look at the variants of those genes and how they are modulated by social interactions. One last quick one. Is there anything comparable to parentage in birds? So this adjustment to the young in the way that you sing? That is a good question. And in that interaction with the family, didn't seem to be much of that going on. It seems that mostly the juvenile is more passively observing the interactions. But I have to say, I probably don't know enough to give you a fully informed answer. But there are situations where possibly if the parents are in a nesting position, that may affect learning. But on the other hand, these are already fledglings, those birds. So the early phase is the more sensory phase, but when they are actually practicing, they're already out of the nest. So there's a lot of interactions going on and so forth. So it's a complex situation. Yeah.
All right, with that, let's thank all the speakers for this round.